Hi everyone, my name is Mark Hughes. I'm the Chief Medical Officer at Cooper Genomics. Most of my career has been spent in academia in multiple universities in the United States. And today I've been asked to speak to you about misconceptions in PGT-M, which is for Mendelian or monogenic testing of embryos, a technology that has now been around for 30 years. It's hard to believe, but true. So we're going to talk a little bit about what this is. Monogenic single gene disorders. We're going to talk about how it screens for them and that it's for couples at high genetic risk of having a child with a disease. Many of these couples don't have infertility. It's going to reduce their risk of having the disease. It requires that we develop a personalized test design. I'm going to explain that to you. So we all carry lots of bad genes. We got some of them from our dad and some of them from our mom, and we really have no idea that we even have them mostly. The good Lord and Mother Nature gave us 2 copies of every gene, and usually the bad ones we got from our dad are not matched on the other chromosome from the gene from our mother, and vice versa, unless our parents were blood related, which is of course why we have rules about that. So we blissfully go through life having no idea we carry it, and then out of the blue we want to have a child with someone who out of the blue has a mutation in the same gene as we do, and now you have a 1 in 4 chance for an autosomal recessive disorder of having a baby with a genetic disease. So we all carry lots of these. You got them from your parents, who got them from your grandparents. They've been in your family for generations, and unless you've had DNA screening or unless you've had a child with a mutation or a family history of it, you don't know. But about 1 in 28 or so of the patients who come to an IVF center actually are at risk of having a child with some genetic disorder, usually autosomal recessive, 1 in 4 chance, or X-linked disorder. And unfortunately in genetics, I don't like to say it, but the truth of the matter is for many of these diseases, it's a field of medicine that's a diagnose and adios, because there isn't really much treatment for most of these conditions. Every cell of your body has the problem with the gene, and there just isn't really good treatments for many of the disorders that are out there, and there are thousands of them. We all know about sickle cell and cystic fibrosis and Huntington and hemophilia, etc., but there are lots of other ones. So when you go through this process of IVF and PGT-M and you do it, your risk goes from 1 in 2 for a dominant disease or 1 in 4 for a recessive disorder, all the way to less than 1 in 20, which is a huge difference for these patients. These are not infertile couples. They're coming to see you because they want to avoid throwing genetic dice, getting pregnant, hoping for the best, and then having weeks of a pregnancy wondering if their baby is affected or not, let alone what in the world they're going to do with the information after they get it. And so PGT-M takes that risk from as high as 1 in 2, 50%, all the way to about 1 in 20 or better. And if you do on that biopsy PGT-M for the disease and PGT-A, which you'll hear about in the next lecture, for chromosome aneuploidy, every embryo you transfer has about a 70% chance of producing a healthy baby. So it's a wonderful change in odds for them. So the beginnings of all of this happened way back in 1990 or so. There was happening around the world. People were trying to do this. I teamed up with Alan Handyside, who many of us would consider the father of preimplantation diagnosis, and he worked at the Hammersmith Hospital, Royal Postgraduate Medical School at the time in London, and worked with Robert Winston, who was the clinician, and I was the geneticist. And we got together and did the world's first case of PGD, preimplantation genetic diagnosis, for a specific genetic disease. In this case, cystic fibrosis. The gene had just been cloned, and we decided to take a common disorder, use the new information on the cloning, and do the first cases of PGT at the time. It's a funny thing because when we sent the abstract into the journal, we said birth of a normal girl after in vitro fertilization and pre-implantation genetic diagnosis, and the journal made us change it to testing. They said, you're not making a diagnosis, you're doing a test. So they forced us to put testing, and here we are all these years later, and the word PGT is no longer accepted. The universal now is called pre-implantation genetic testing, in this case for monogenic disease. So the editors were right way back then. So the technologies have dramatically changed. We're going to go over some of those today. So has the sample, because in those days, most clinics were transferring two pronuclei fertilized eggs because the culture conditions were so terrible back in 1990 compared to today. Then Handyside was able to grow embryos to day 3. We could biopsy them, but we had to get those embryos transferred later that day because growing to day 4 and day 5 was incredibly unlikely to be successful. So that's dramatic changes. And unfortunately, after all these years, I've been around now for 30 years doing this, and after all these years, misconceptions about the technology still exist. That has not changed. So one of those is that, well, DNA sequencing is now pretty routine. We've sequenced the genome, we'll just take the DNA, you just take the biopsy and send it to the lab, they throw it in a sequencer. The sequencing machine chugs along all night long, and voila, you have a molecular answer the next day. Well, that's unfortunately not the way it works. It's more complicated than that. But we do sequencing. It's a different kind of sequencing, and in today's talk, I want to make sure you understand those differences and understand why sequencing of that biopsy you send is not sufficient to do PGT-M accurately. So you have to do something else called karyomapping, and I'm going to talk about that as well. One of the main reasons for this is that the sample you send only has 4, 5, 6 cells in it. It doesn't have millions of cells like an amniocentesis or a CBS or a blood sample or a tissue sample. It only has a few. So as we talk about genome equivalence, there isn't very much DNA in there. And DNA sequencing is very good, but it needs something to start with. And even if you do PCR, it doesn't get you around the problems of starting with such a small amount of material. And we'll talk about that in a second as well. Also, the sequencing that you would do in order to look at a mutation would be incredibly expensive per sample. And if you multiply that times 6 embryos, it would be very expensive for the couple. So for those reasons, we use a different kind of sequencing. So here's a stretch of DNA, and we're going to talk about sequencing read depth for a second. So DNA has a reading side, it's called the 5' end, and it reads along all these A, T's, G's, and C's. And I'm going to just parenthetically tell you a bit of fun here. So it turns out that if you were to type the human genome from one of those biopsy cells, not 5 of them, just one of them, in 12-pitch font in your computer, it would stretch 5,400 miles, or about 9,000 kilometers long. And if you could type at 75 words a minute, it would take you, at 8 hours a day of typing, it would take you over 50 years to type out the 6 billion letters in the human genome. And you'd have a book with about 1 million pages in it, or roughly 200 novels, each with 5,000 pages in. You can think of those novels like chromosomes, and they all have information about you, but this is one hell of a big book. And so here's just a tiny little section of some DNA in chromosome 4, and I put a mutation in the middle of this to have you appreciate that out of all these billions of letters of DNA, one little letter base change is all that it takes to cause a disease. Now, DNA sequencing machines, what they do is they don't work like people think, where the enzyme jumps onto the DNA. This is called a DNA polymerase. It hops on and it's like a locomotive train and starts running down the racetrack, reading the DNA and keep on going and going and going all the way through until it falls off on the other end. It would be nice if that was the case, and in fact there's some technologies on the horizon that look like that might be possible with some reliability. But at the moment, that's not how sequencers work. Instead, what sequencers do is they sequence DNA in very small little pieces. First of all, the enzyme doesn't stay on a long way. And secondly, we break the DNA up from those little biopsy that you send. We break it up into pieces. And so the sequencer just sequences that little bit. And then the computer looks at that sequence and says, aha, that is a phrase in book number 4, or no, that's a sentence in book number 6 or 21 or whatever. And so you get these little tiny pieces of DNA that go along. And there might be one here, and then there might be another one down here, and then there might be one up there. And the longer you run the machine, the more of these little sequences that you get. And so you can get a lot of information, but the longer you run it, the more reagents you needed and the more expensive the test is. So in general, what we do is we have low, mid, and high resolution depth DNA sequencing. So this would be considered like a low or medium depth. And this is perfect for just kind of getting a GPS logistics on your sample. So it's not looking at, look, all this DNA wasn't even sequenced and all this DNA wasn't sequenced, but enough of it has been sequenced that it gives you a bar in a barcode that tells you, aha, these pieces of DNA belong to, I'll keep picking on chromosome 4. And that's useful in things like non-invasive prenatal testing early in pregnancy. It's useful in PGTA, which the next speaker is going to talk to you about. So that's all great. But if you notice, it didn't pick up the mutation, and it would have randomly perhaps picked it up. So sequencing using these machines would be unreliable. And you remember, you've also got another copy of this DNA from the other parent that you have to be seeing, and you have to be seeing them both at the same time. So low and mid-level sequencing read depths are okay for a lot of things, but not okay for PGT. So indeed, it can tell us that this is on chromosome 4, but that's kind of about it. So let's focus in on our mutation, because of all of those nucleotide changes, all those nucleotides in the genome, only this one is the one we're really, really worried about. Now, if we were to spend the effort, we could do deep sequencing. So this would be that same sort of sequence. It would just keep right on going, and we're going to just keep the machine running. And as it runs, it makes more and more copies of this and just keeps right on going. And the deeper you sequence, the more confidence you have in every DNA nucleotide that you have. And if you notice here, you've got a lot of overlap because you ran it a long time. So some regions, like say this A here, was sequenced twice in that particular run, where another sequence over here has been sequenced 4 times, where this one over here has been sequenced twice. And in this example, our mutation was hit 3 times, so we start to get confidence that it's correct. But as I said, this is expensive, and when you're doing this for 6 or 8 embryo samples, it's too expensive for patients to reasonably want to do this. So this is used for mutation hunting. You've got a child in the nursery and no one can figure out what it is. You do deep whole genome sequencing and you look and see if you go hunting and see if you can find a mutation in a gene that suddenly gives you an aha moment about what's clinically wrong with this child. So clinical dilemmas, it's very much used for, but it's not used in PGT at all. So what is? Well, remember now, in PGT-M, we know precisely what we're looking for. Way before the patient begins IVF, we know the disease, we know the inheritance, we know the family history, and we know the exact mutation. So we can create a test with this information that functions like a guided missile. It doesn't sequence over on a different chromosome from where the gene is that's of concern. And that is helpful in keeping the costs down and actually increasing the accuracy. So here we have our mutation again in this DNA, and what we're going to do is we're going to sequence specifically on that mutation. So we take some of the DNA out in front on the 5' end of the mutation and down on the 3' end of the mutation, and we make little artificial pieces of DNA that will allow a PCR machine to run back and forth and make us millions of copies of the thing that we're particularly interested in. And that's what's been done now for a very long time in PGTM. The problem is that's not good enough either in today's world. We really, really want to be accurate. And with 5 cells, you better be sure that you saw that A if it existed, and that you saw the DNA letter from the other parent if it exists. And so what we do is the Genome Lab creates a custom family test that only looks at these particular regions. So this is our sequencing region, and now we can't forget that we've got the other parent. So let's say that this DNA running along here is mom's DNA, and this is the mutation that mom carries, this A. And every so often she's got a letter in her DNA that's different. Your DNA is different from everybody else's unless you have an identical twin, and that's how the police can figure out if you've been at the scene of the crime, or they can do forensics or population migrations around the world. They use these polymorphisms, differences, multiple morphisms, changes of the DNA, and they're single letters, so they're called SNPs. So this is mom's DNA in this example. So let's look at dad's DNA, and at some of these SNPs, he's got a different letter. All the rest of these letters are the same in his sequence, but he's got a different one, and that's great because each one of these is like a bar and a barcode and like on a soup can, and we can tell the difference between mom's strand of DNA and dad's strand of DNA, not by looking at these letters, but by looking at these polymorphic differences that they have. And by doing that, we can be sure when we do the test that we actually are looking at a strand of DNA in that embryo biopsy from both gamete donors. You don't want to have only moms and see only A and say, well, dad must have an A, when in fact what's happened is the sample has maternal cell contamination, for example, or the sequencing reaction failed to see dad's DNA, for example. So instead of just having one data point, the A or not, we've got multiple data points that give us additional confidence that that biopsied sample actually has what we want it to have. So mom's and dad's DNA are different, and those differences allow us to follow both strands of DNA in each of the cells from the biopsy you sent, and we can tell that we didn't miss a G, it actually was there, and that G is associated with these other polymorphisms on the upper level. So because we have so many of them, we can follow them along. So here's dad's DNA, let's say, and here's mom's DNA. This is the centromere of the chromosome, and this is out to the telomere, and here are the two, and one of those individuals has a mutation in the gene. And we can follow those little SNPs out in front of the gene, through the gene, and out the back end of the gene for both times. So we absolutely know that this is the strand of DNA that's associated with this, even though these are little tiny pieces of DNA. Each one of these are little pieces that we're sequencing. And this all happens, including recombinations or genetic crossovers, if you remember from your basic biology, because way back in meiosis, when that egg is still in the ovary, it is doing a recombination between grandma and grandpa's chromosome that they gave to your parent. And so we get a mixing, like a deck of cards, we get a mixing of the chromosomes across our family tree, and we can follow the pink, which is the normal one here, and we can see the crossover where it goes to the other one in that particular embryo. Gives us much, much more confidence. This whole process is called karyomapping, which you've probably heard along. Okay, so to summarize then, what we've got is we've got dad, we've got a mutation, we've got mom, we've got a mutation, we've got polymorphisms that we follow all the way along, and that's why we ask, and laboratories ask, for not only the embryo samples in PGT-M, but also the parents, ideally the grandparents if they exist, a child if they've had one, or a sibling that happens to have the genetic disease. It allows us to link up the SNPs with which strand of DNA has the mutation and which one doesn't. We analyze those SNP samples surrounding the gene, making it a very powerful test and telling the difference. That whole process is called linkage analysis. So the other misconception in PGT-M is, well, all right, since sequencing is so quite routine, all the embryology lab has to do is perform the biopsy and then ship it to the lab and they'll do the test. Well, I hope by now in this talk you realize that that's not the case. It is the case in PGT-A, but it isn't in PGT-M, where you have to do some homework beforehand. So you need to be communicating with the laboratory. You don't just send samples. Another misconception is that we can do this for all genetic diseases. But actually, this misconception isn't so far off because we almost can. So it's almost true if we know the molecular basis of the disease. So if you call and say, can you do PGT-M for autism? The answer is no. Why? Because there's a lot of genes that have been loosely associated with autism, but none specifically. And so we don't have anything to send our missile to. We don't have a target in which to do our homework on. So there are diseases, polygenic diseases, that only happen with multiple gene defects. And others, but by and large, if we know what's causing the disease, what the mutation is, and that can be found fairly straightforward in the family, then the test can be designed. And that's true because it's been shown to be true even because over 6,000 different single gene disorders have now been tested for, including even things like inherited cancers. Most cancers are not inherited, only about 10% are. But breast and ovarian cancer through the BRCA mutations particularly, PGT-M has been done quite regularly for that. Cystic fibrosis, very common. Fragile X, spinal muscular atrophy, Huntington disease, sickle cell. Those are the most common ones in the United States and Europe. But for example, if you're from South America, Mexico, etc., or from Asia, we wouldn't be thinking about cystic fibrosis at all. And that's a rarer condition because the gene isn't prevalent in that population. So it really depends on where the family lives. And because we perform these tests all around the world, we've had to develop the tests for many, many families. We cannot test for disorders that are associated with mutations in mitochondria. And it would take me a while to explain to you why that is. Just believe me when you say that's not that we can't do that. And we can't do it for genomic imprinting either. But truthfully, it's not likely any of you guys are going to see any mitochondrial or genomic imprinting disorders anyway. Do you always need a family member in order to build a PGT test? Well, traditionally, yes. But technology and innovation has moved on, and now I can say not always. So let's take this example. We've got Dad here, and let's pretend that he has a dominant disease. He's got a mutation in one of his 2 chromosomes. Let's say he got it from his mother. And he wants to have children with his mate. And maybe he's been adopted, so we don't know anything about the previous generation. Or maybe they've died, or they're estranged, or he wants to keep this all private and not tell them. Or maybe he doesn't have a brother or sister with the same disease and they don't have any children with it. Well, then how are you going to connect the SNP strand of DNA with the mutation strand of DNA if you don't have anyone other than that individual's 2 chromosomes to link it to? How do you do that? So if we look at that more in a picture, we've got Dad, we're going to give him a G and a T, and we're going to call the T the mutation. And if he got a 50-50 chance, if he gives the T in his sperm, that embryo, that offspring will have the same disease. If he gives the G, then the baby won't have it. Mom has got 2 different letters and her 2 copies of the gene, one from each of her parents, and both of those are normal. So we don't want to just do sequencing because we want to be sure that if the T is present in the embryo, we actually see it, and that it isn't just we think there's 2 G's or something. And so if we look at that the way I showed you in the previous slide, we want to be able to see that T, and we want to be able to see the linkage, the SNPs on each side of that gene for both mom and dad. But how in the hell are we going to do that if we don't have anybody else in the family? Well, what we can do is we can look at their embryos, and we can pretend for a little bit that their embryos are 6 children in the family already sitting around the dinner table, and now they want to have more. So we're going to test these embryos. We're going to do DNA sequencing and SNP analysis and solve the puzzle about how the SNPs and the mutation are linked to one another after we collect the data instead of before. So this isn't ideal, but it works. It works quite well. So we look at the first embryo and we see a G from dad, that's normal, and mom has to give something, she gives an A, and we know that embryo has that set of SNPs. We don't know anything yet about the mutation, but in the next embryo, we know that the T is present and it's associated with a different set of SNPs, a different haploblock, a different allele strand, a different barcode or fingerprint. Any of those words work. And then we look at the next embryo and we see the same polymorphisms, and we work our way down the row. And every single time that we see a T in our little missile-guided sequencing, we also see the same SNPs that are associated with it. And now if we've done that several times in several embryos, we have linked the SNP set with the mutation. And then we can have a lot of confidence that not only is our sequencing working correctly in this particular family, but also we're getting consistent result across the entire group of embryos. So this is the kind of work you have to do in order to be sure. And you want to be sure, of course, in these cases. So we can develop a test. It's not ideal. We'd like to have 3 affected embryos in the cohort in order to do this, but it's very doable. It's mostly suitable for de novo mutations. He's the only guy in the family with it, and he doesn't have any way of connecting the SNP to the mutation. Maybe he's adopted, as I said, or he has an unstable family, whatever. We also hear the misconception that PGTM isn't suitable for microdeletions. This is simply not true. If you can follow polymorphic markers across a piece of DNA and you can show that those markers are missing in samples that have a microdeletion, that the fingerprint is half there during the microdeletion part of the sequencing, then you can molecularly brand that strand of DNA as normal or microdeleted and follow it in the embryos. And so this is done all the time, so anybody that tells you it's not possible, it's because their technology isn't cutting edge. So in summary, we're able to accept the most complex disorders. We've been doing this for 30 years. We've done it for thousands of diseases. We've got lots of experience and expertise. We can run PGT-M and A simultaneously, which is quite nice, the same biopsy, and run them both simultaneously. And we have all of the certifications that one would want around the world to do this under the correct auspices of government at all and regulatory group approvals. So with that, I think I'll just stop for a moment. We'll take questions later, and I'll pass it on over to David, who's going to tell us all about how we use these technologies to look at aneuploidy in chromosomes. David?
Hi, and thank you, Marcus, for that great presentation on the misconceptions in PGT-M, and for Tony for introducing me earlier. So now I'm going to just quickly take over and talk about some of the misconceptions in PGT-A. So let's start with the PGT-A overview. So PGT-A is pre-implantation genetic testing for aneuploidy. Now previously this was called PGS, or pre-implantation genetic screening, or CCS, comprehensive chromosome screening. And the good news for those was they actually had the word screening in the name. And that's very important because it is a screening test, not a diagnostic test. So it screens for chromosome abnormalities. And for meiotic errors, i.e. errors originating in the oocytes, it has a very high accuracy of around 97 or greater %. So if the trophectoderm biopsy says it's euploid, the inner cell mass is euploid in 97 or greater cases, and the same for aneuploidy. So it is a very good screening test. And the other good thing for PGTA is it is an option for all IVF patients. It doesn't require any kind of personalised test prep or medical history. It is an option. Whether you choose to offer that option to the patients is up to the patient and clinicians in discussion. But it does improve IVF success rates, and especially for those in the over 35 bracket. So PGTA has been shown to improve IVF outcomes. So one of the first studies showed that it mitigates the effect of maternal age. And by that mean that with a good embryo, a euploid embryo, pretty much have the same success rate per embryo transfer with an advanced maternal age as a young female patient. And that is because it reduces miscarriage rates. So it really brings down the miscarriage rate to background levels. So this does not eliminate miscarriage, and it shouldn't be seen as a miscarriage elimination, but it certainly reduces it. And it also increases the implantation rates per embryo transfer. So combining increasing implantation rates with reducing miscarriage rates, we are increasing our live birth rates per transfer. By using PGTA to prioritise embryo transfer, then we also have reduced time to pregnancy. And what it has also done is enable much more confident single embryo transfer, especially in the more advanced maternal age, where there was always the temptation to try and increase the amount of embryos put back to try and ensure at least one of them was a successful pregnancy. We don't need to do that with PGTA. So what is equally important is the accuracy of the test. So what we have done recently is launch PGT-AI2, and you can find out more details on our YouTube channel. I'm not going to go through the whole PGT-AI2 presentation today, but now with the increased sequencing, that we can now do much more accurate assessment for the ploidy status and mosaic status of the embryo. And with parental DNA, for the highest accuracy, we can detect all forms of ploidy, so female haploid, female triploid. We can test whether that aneuploidy is from maternal or paternal origin, but we can also do the 2PN validation. So if it's not haploid, it's not triploid, it's diploid, and we can see the heterozygosity and show that it's 2PN. And this feeds into the data quality. So I've been in PGTA now for 12 years. I've seen a real explosion in the technology from where we started with polar bodies, 24 shore chromosomal arrays through to trophectoderm now and 4 million paired end sequencing reads. But all the way through this, transfer prioritization decisions were based on the PGTA results in part. What we now have using AI is the ability to use high-power statistical analysis from a baseline which includes live birth and sustained pregnancies and also known aneuploidy embryos that have been shown to be completely aneuploid by secondary methods. And this allows us to baseline all of our decisions for that embryo being tested using high-power statistics against those matched profiles. And because for each embryo tested we're using these high-power statistics, we remove the user bias. There is no, did I see this yesterday? Does it look like one that I saw 2 weeks ago? How do we report that? It's all computer-based. And that gives us great repeatability. And because we still track outcome data, we are now able to build incremental improvements. So the more data we have to feed into the known outcomes, the better that will become. And this is the great advantage of artificial intelligence. So even with PGT-AI 2.0, we have a limitation of detection ability, and it's important that the patients and the clinicians understand the limitations of the PGT-A technology. So PGT-AI 2.0 can detect the whole chromosome changes, which have always been done. But now we can get down to 5 megabases with high accuracy, and whilst we're talking about PGT-A today, this is obviously very important for the PGT-SR, the structural rearrangements, which often involve small telomeric segments. So we can do the mosaic changes, as I've said, and the parent of origin. What this doesn't say is what we can't detect, and this is equally important. So we can't detect anything below 5 megabases, and these are rare, but they can happen, and sometimes they are clinically relevant. And I would just pull out one as a highlight, which is DeGeorge syndrome. This is a microdeletion at chromosome 22q11.2, and is quite often 250 kb or smaller. It's usually de novo, so no inheritance, and this would slip below the detection threshold. So it's very important that we understand when talking to patients that PGT-A is not going to detect every possible chromosomal abnormality and genomic abnormality in that embryo. So, can an abnormal embryo self-correct? There has been a phrase, mosaic embryos self-correct, banded around, but is this actually true? So what do we mean by self-correction? A meiotic error resulting in a normal embryo, or a mitotic error resulting in a mosaic embryo resulting in a healthy live birth, or a combination of the two? So to have a meiotic error, then there would have to be a non-disjunction error leading to either a monosomy chromosome or a triploid chromosome. And if it was a triploid, so say trisomy 15, then there would have to be a successful removal of the extra chromosome from all cells, and the remaining 2 are from one parent. So clearly, theoretically possible. However, in a UPD that we see in live births, even from a normal pregnancy, we can't track whether that is what happened or whether it was simply that one of the oocytes had both copies of that chromosome, and the corresponding oocyte didn't have it at all, and it's just gone on and formed a pregnancy. So there is no real way that this has been managed to show the mechanism for ending up as a UPD. If it was a monosomy, then the cell would have to undergo duplication of that chromosome prior to cell division so that it's populated throughout all of the cells and all of the embryo. And again, we haven't really got any evidence for that. Now, mosaic embryo outcomes, yes, we know there have been reports that mosaic embryos have been transferred and they have resulted in the main in what appears to be a healthy live birth. So we do know they have reduced implantation potential, they do have higher miscarriage, but some make it to live birth. So how is that possible? Is it undergoing self-correction? Well, what we do know is that with imaging, is these embryos which are mosaic have a higher rate of cell death. And therefore it would appear that the embryo is in some way able to have the abnormal cells, they undergo apoptosis, leaving the euploid as the dominant or only cells left. But as that mosaic load increases, the amount of cell death required for that to happen becomes detrimental to the embryo survival. So is that self-correction? And at one level you could argue it is, but really it seems that abnormal cells are more likely to undergo apoptosis than euploid cells. So PGT-A guarantees a healthy baby? No. Much like PGT-M, PGT-A does not guarantee a healthy child. As I stated earlier, it is a screening test. Equally, you can have confined placental mosaicism and other conditions where the trophectoderm biopsy, which ultimately goes on to form the placenta, is euploid, the inner cell mass is aneuploid in some way. And so society recommendations is that all women, even after PGTA, should undergo the usual prenatal screening tests, including the option of an invasive diagnostic test during pregnancy. So in no way does PGTA guarantee a healthy baby. So one of the things where there's been a lot of discussion recently is the non-invasive PGT. So quickly, where are we with that? So basically there have been quite a few papers published over the last few years, and overall it shows that the embryology is critically important for success. So you absolutely require single embryo culture from day 3 or day 4. So if you're a clinic who believes in group culture, this is obviously a potential issue. You also have to wash the embryos through various, some say 3, some say 5, media droplets at day 3 or day 4. Again, if you're doing things like time lapse or if you're doing uninterrupted culture, this could prove a barrier for non-invasive. The low media droplets that have to be probably smaller than 20 microliters. For some that's fine, for others it may raise questions depending on their incubators. But the one thing that all the papers have shown is to get some really good results, you need to have the embryos in that media culture for at least 48 hours. So across all these studies, the overall concordance to invasive PGTA is variable, and now PGTA is so well established and people have outcome data for euploids and aneuploids and mosaics. This is really driving the question as to how applicable are the results you get for non-invasive PGTA and how close in concordance do they need to be. And there are still issues of maternal contamination to be resolved. So after fertilization, you've got the polar bodies being excluded, you've got cumulus cells potentially releasing DNA into the media, and it has been shown that a lot of the false positives are actually female euploid, which is actually the maternal cell contamination and not the embryo. So in summary, PGTA remains the best available tool for assessing the genomic status of the embryo. It has been reported numerous times that euploid embryos have the highest success rates and that mosaics have a lower implantation potential. PGTA will continue to develop with more sequencing, more data and more accurate results. However, it is just one of the tools in the embryo selection portfolio. So it is not a catch-all. There is also morphology and various other aspects which will almost certainly influence the outcome of those embryos.